Hello everyone, this is chapter number 18, Body Fluid and Circulation. In this chapter, I'll be making 5 videos. This is video number 1. And uh, in this video, let us first understand what is circulation. Circulation is the transport of nutrients, oxygen, carbon dioxide, and excretory products to the concerned tissue or organs. For circulation, simple organisms like sponges and coelenterates use water from their surrounding. Complex organisms use body fluids like blood and lymph for circulation. Now let us see to the types of circulatory system. Basically, there are two types of circulatory system, open and closed type. Open circulatory system, the blood is pumped by the heart and it passes through the large vessels into an open space called sinus. You can see over here, followed my cursor, this is the heart, and from the heart it comes to the vessels, and from the vessel it comes into the large space which is known as sinus. And this you generally see in arthropods and mollusks. Then comes this closed circulatory system. In the closed circulatory system, you can see over here the heart. The blood is flowing from the heart to the vessel and back from vessel to the heart. It does not come out. And it is more advantageous as the flow of fluid can be precisely regulated. And you see it in annelids and all chordates. Then let us see the different types of hearts in the vertebrates. Now, if you talk about the different types of hearts in the vertebrates, they are in fish, you see two-chambered heart. In amphibians, it is three-chambered heart. In reptiles, except for crocodiles, it is again three-chambered heart. And in crocodile, birds, and mammal, it is four-chambered heart. In amphibians and reptiles, both have three-chambered heart, except for crocodiles. But what is the difference? In amphibians, it has two atria and one ventricle. In reptiles, it is three chambered, but the ventricle is incomplete partition. Uh, and that's the difference between the two. In fish, it is two chambered, one atria and one ventricle. Talking about the patterns of circulation in vertebrate, the patterns of circulation, this of three types: single circulation, incomplete double circulation, and double circulation. Talking about each one of them, single circulation, it is seen in fish. Here, the heart is known as the venous heart because it receives impure blood. And if you see to the circulation pathway, the pathway, then it is deoxygenated blood goes to the heart. From the heart, it goes to the gills. In the gills, it gets oxygenated. Then it goes to different parts of the body. And from the body, the deoxygenated blood moves to the heart. Incomplete double circulation is seen in amphibians and reptiles. In this, the left atria receives oxygenated blood from the gills, lungs, or skin, and the right atria receives deoxygenated blood from the body. Both the deoxygenated and the oxygenated blood comes into the ventricle where it gets mixed, and then this mixed blood is passed to different parts of the body. Talking about double circulation, which we see in birds and mammals, which we will be studying in detail, and there is no mixing of blood. You can see the right side receives the right side receives deoxygenated blood from all over the body, the right atrium, then it comes into the right ventricle. Then from the right ventricle, the deoxygenated blood through the pulmonary artery goes into the lungs where it gets oxygenated. And the oxygenated blood then comes on the left hand side. And then with the help of the aorta, it goes to different parts of the body. So there is no mixing of blood in this. Now let's study about the human circulatory system. In the human circulatory system, we'll be talking about blood, blood vessels, and heart. Basically, the human circulatory system is divided into two systems. One is blood vascular system and another is lymphatic system. Talking about lymphatic system, it has lymph vessels, and lymph nodes. But blood vascular system has three things: heart, blood, and blood vessels. Blood is divided into two parts: blood plasma and formed elements. Formed elements are of three types: RBC, WBC, and platelets. WBC are of two types: granulocyte and agranulocyte. Granulocytes are of three types: neutrophil, eosinophil, and basophil. Agranulocyte is of two types: lymphocyte and monocyte. Lymphocyte again is of two types: B lymphocyte and T lymphocyte. So we'll be talking about it in detail in the coming slides. Blood vessels are of three types: artery, veins, and capillaries. Now let's talk about the blood. The first part of the blood, that is the composition of the blood. Blood is made up of two things: plasma and formed element. 55% of the blood is plasma, that is straw colored and it's slightly alkaline. The pH is 7.4 and it forms the fluid matrix. Now, the inside in this blood plasma, it includes about 45% of formed elements. These formed elements includes the different cells, that is the RBC, WBC, and platelets together is known as the formed elements. Now, let's talk about plasma in detail, constituent of plasma and its function. It's 90 to 92% of blood plasma is made up of water, and that's why it's a good solvent. In the plasma is present 6 to 8% of plasma proteins like fibrinogen, globin, and albumin. Fibrinogen, that takes part in blood coagulation. Globulin acts as an antibody for the defense of the body, and albumin as osmotic balance and regulate the blood pressure. Then the plasma also contains glucose, amino acid, lipids, cholesterol, some inorganic ions like sodium ion, magnesium ion, chloride ion, bicarbonate ion, calcium ion, some gases like carbon dioxide, oxygen, nitrogen, etc. Now, plasma without the clotting factor, that is, without the platelets and the clotting factor, is known as serum. Now, let's talk about the formed elements. Learn about the formed elements. There are three types: RBC, WBC, and platelets. Together is known as the formed elements. Now, let's see one by one each of them. Let's see first RBC, which is known as erythrocyte. If you see to the color, it's red in color due to the hemoglobin pigment. Normal HB level in blood should be 12 to 16 gram per 100 milliliter. Lifespan is 120 days. The count of RBC is 5 to 5.5 millions per cubic millimeter. They are formed in the red bone marrow of long bones. Structure, it is biconcave in shape. No nucleus is present. Mitochondria is not present, as well as Golgi apparatus is also absent. Basically, what is the function of RBC? It transports carbon dioxide and oxygen. You can see over here the structure of an RBC. It's biconcave in shape, and the worn out RBCs, the dead RBCs, they are destroyed in the spleen, and that's why the spleen is also known as graveyard of RBC. Now let's talk about WBC or leukocyte. Basically, leukocytes or WBC are colorless. The lifespan is 1 to 15 days. Its count is much less than RBC. It's only 6,000 to 8,000 per cubic millimeter. They are also formed in bone marrow, lymph glands, as well as spleen, but they are nucleated and they are of different type. And their main function is to fight against the germs. Talking about the different types of RBC, as I told you, we had just now seen in the flowchart, they are of two types: granulocyte and agranulocyte. Granulocytes are of three types: neutrophil, eosinophil, and basophil. Neutrophil is 60 to 65%, and they are phagocytosis in nature. They are also known as the soldiers of the body. Eosinophil is 2 to 3%, and they resist infection. They cause allergic reactions. Basophil is 0.5 to 1%. It secretes histamine, serotonin, heparin, and cause inflammatory reactions. Talking about agranulocytes, they are of two types, monocyte and lymphocyte. Monocytes, they are the largest WBC and it forms about 6 to 8%, and they are phagocytosis in nature. Lymphocytes, they are the smallest WBCs, but their nucleus is largest. They are of two types, B lymphocyte and T lymphocyte. They form total of about 20 to 25%, and their function is immune responses of the body as well as secrete antibodies. Now let's come to the third formed elements, that is the platelets. And just let us see, which is also known as thrombocytes. What are the features of the platelets? The colorless. Lifespan is 7 days. Count is 1.5 to 3.5 lakhs per meter cube. They are formed in megakaryocytes in the bone marrow. Megakaryocytes are the cells which forms the platelets. If you see to the structure, they are non-nucleated cell fragments. You can just see these are non-nucleated cell fragments. These are the inactivated platelets and these are the activated platelets. And the platelets, we all know their function is blood clotting. Basically, if we talk about blood clotting or blood coagulation, it is a mechanism for homeostasis. Homeostasis to means to maintain the internal environment. Otherwise, blood loss is going to take place. And it prevents the blood loss, these platelets. It forms a mesh-like structure, and those whose platelets problem is there or blood clotting does not take place, a disease known as hemophilia or bleeder's disease. Now let us study the different steps of coagulation of blood. Now, whenever injury takes place, at the site of the injury, the clumped platelets and tissues are released. These clumped platelets and tissues, they release thromboplastin. This thromboplastin converts prothrombokinase into thrombokinase. In the blood, already prothrombokinase is present. That means thrombokinase in inactive form is present. Now, this prothrombokinase acts as an enzyme and it converts prothrombin into thrombin in presence of calcium ion. Now, this thrombin converts fibrinogen, which is already present in the blood in the soluble form, into fibrin, which is insoluble. Now, this fibrin, which is insoluble, traps the dead and the damaged formed elements and forms a clot. So, this is the steps of coagulation of blood. Children, if the question comes for coagulation of blood and if you write these steps, the whole thing, you get full marks in the boards. You can see over here also the injury as a result bleeding is taking place, and then the platelets get activated and fibrin is being formed. That fibrin traps these platelets, the damaged platelets, and the blood gets coagulated. Now next, let us talk about the blood grouping. The blood group in human, the blood group was discovered by Karl Landsteiner, and it includes the ABO grouping and the RH grouping. Talking about the ABO grouping, it is based on the presence or absence of two surface antigens. These are the chemicals that induce the immune response, and these antigens are present on the RBC, and they are named as antigen A and antigen B. You can see over here, antigen A and antigen B. Similarly, plasma contains two antibodies. They are known as anti-A and anti-B. So antigens are present on the RBC, named as antigen A and antigen B. And the antibodies named as anti-A and anti-B is present in the plasma. Now let's see to the ABO blood grouping. There are four types of blood group A, B, AB, and O. Blood group A contains antigen A, anti B. The antibody present is anti B. The blood group A can donate blood to person having blood group A or AB and can receive from A and O. Same way, person having blood group B has got antigen B, antibody anti A is present. It can donate blood to people having blood group B and AB, but can receive from B and O. Same way, the person having blood group AB has both the antigens, that is antigen A and antigen B on the RBC, but they do not have any antibodies in the plasma. They can donate blood only to AB, but can receive from A, B, AB, and O. So they are universal acceptor. The last blood group, that is blood group O, does not have any antigen on the RBC. Rather, they have both the antibodies in the plasma. They can donate blood to person having blood group A, B, AB, and O. So they are universal donor but cannot receive blood from anyone except for O. Now, when antigen A, if a person who is having blood group A has antigen A and suppose blood to him, some other blood group is given, like blood group B is given. Now, the blood group B has got anti B. So, what happens? Anti A. The blood group B has got anti A. So, what happens? Antigen A reacts with anti A. Same way, vice versa, if somebody is having blood group B, and to him, if you give blood group A, then that blood group A has anti B, so it undergoes reaction. And this mixing of blood with interactive antigen and antibody causes clumping or agglutination of the RBC. As I just now told you, blood group O is universal donor and AB is universal acceptor. Now let us talk about RH grouping. RH, rhesus factor, that is an antigen that is found in RBC. There are two types of person. One is RH positive and another is RH negative. RH positive means there is RH factor is present on the RBC, and RH negative means the RH factor is absent. 80% of humans are RH positive. Now, this anti-RH antibodies are not naturally found. So, RH negative person can receive RH positive blood only once. But it causes the development of anti-RH antibody in the blood. So, the second transfusion of the RH positive blood causes agglutination. So, you can see over here, he is RH positive. RH positive can give to RH positive. RH positive can not give to RH negative. He can give once, but if he gives once, then the second time, whenever he gives the blood, then agglutination will take place. Same way, if a person is Rh negative, can give to both Rh positive as well as Rh negative, no problem in that. Now, this Rh positive and Rh negative, there isn't, the problem takes place in pregnant women. That is known as Rh incompatibility between Rh negative blood of a pregnant mother and Rh positive blood of a fetus. If the mother is Rh negative and the father is Rh positive, then the fetus is going to be Rh positive. Then the Rh antigen do not get mixed up with the maternal blood in the first pregnancy because placenta separates the two bloods. But at the time of delivery, there is a possibility of exposure of maternal blood to small amount of Rh positive blood from the fetus. Now what happens? This induces the formation of Rh antibodies in the maternal blood. And then, in case of a subsequent pregnancy, the Rh antibody from the mother leaks into the female RBC, and this is fatal to fetus and causes severe anemia and jaundice to the baby. This condition is known as erythroblastosis fetalis. I'll show it again to you. See, the mother is, now look here at this diagram and follow the cursor. The mother is Rh negative. Father maybe will be must have been Rh positive. So 100% the fetus is Rh positive. So there is no mixing of the blood when the first baby is growing. But at the time of delivery, uh, some of the Rh positive comes in contact with the mother's blood at the time of delivery. Then what happens? In the second time, the mother develops anti-Rh. So during the second pregnancy, this anti-Rh, this yellow color boxes, they come in, and then the fetus blood undergoes agglutination and the fetus dies. This is known as erythroblastosis fetalis. Erythroblastosis fetalis nowadays can be avoided by administering anti-Rh antibody to the mother immediately after the delivery of the first child. Now over here, I finish the first video. Then next video will be made about the different types of arteries and human structure of human heart. Thank you.